Hi, everyone, and welcome to your introduction to leukemias and white blood cell abnormalities. So this is the start of the leukemia series. Before we get into leukemias part 1, 2, and 3, we talk about some methodologies, some white blood cell methodologies. So you have recorded lectures on cytogenetics, molecular diagnostics, and also on flow cytometry. Okay, so let's begin. Here are your objectives. This lecture was originally prepared by Dr. Elaine Cohane. She was a associate professor at Rutgers Medical Lab Science program. And I have permission to use her previously prepared slides. Okay, so let's start. So in terms of leukemias, most leukemic or most hematologic malignancies or malignancies of the hematopoietic system are acquired, which means that most patients are not born with it, but then at some point in life, they acquired this type of deficiency. For most leukocyte neoplasms, the causes are directly related to the development of the malignancies, and the cause is unknown. Some malignancies have been associated with exposure to organic solvents such as benzene or exposure to radiation, perhaps previous exposure to alkylating agents like chemotherapy. Some malignancies have been associated with viral infections. For example, there are two types of lymphoid malignancies in which viruses have been implicated in the etiology. Epstein-Barr virus, EBV, it has been implicated in the development of Burkitt's non-Hodgkin's lymphoma. And the other example is the human T-cell lymphotrophic virus type 1, the HTLV1, which has been identified to be the cause of adult T-cell lymphoma. So mutations and malignant transformations found in leukocyte neoplasms can lead to one or more of the following: dysregulated growth or uncontrolled proliferation due to disruption in cell signaling, increased capacity for self-renewal, resistance or circumvention or avoidance of apoptosis, dysregulation of the cell cycle, a block in differentiation or a maturation arrest, decreased expression of tumor suppressor genes, epigenetic silencing of transcription of essential cellular genes, and or mutations in DNA repair genes. So now let's talk about some of the classifications of leukemias. A leukemia originates in the bone marrow and peripheral blood, or the peripheral blood. The lymphomas originate in lymphoid tissue. Hematopoietic malignancies present in two ways, either acute or chronic. Acute leukocyte neoplasms implicate a rapid onset of disease, which means they progress rapidly and they can be fatal in weeks or months. Chronic leukemias, on the other hand, present with an insidious and slow onset and are usually less aggressive, and the survival is longer. The subtype in the leukemias are classified as either lymphocytic or non-lymphocytic. There are two classification schemes that we use for leukocyte neoplasms. In the 1970s and the 1980s, the French-American-British, the FAB classification scheme was used, and it was relied heavily on blood cell morphology and histological stain preparations to tell the difference between lymphoid and myeloid malignancies. Although these types of criteria are still being used, pathologists are now moving towards diagnosing leukocyte neoplasms based on recurrent chromosomal abnormalities, and that is presented by new criteria that has been displayed by the World Health Organization. It was first described in 2001, and then it was later revised in 2008 and then in 2016. And this is the newer classification scheme for nearly all tumors of the hematopoietic and lymphoid tissue that mostly classifies malignancies based on recurrent chromosomal and genetic abnormalities. More than 50% of leukemias and lymphomas have detectable genetic abnormalities. More than 90% of all the acute lymphocytic leukemias, the ALLs, have detectable genetic abnormalities. The types of mutations seen in leukocyte neoplasms are some of the following. Translocations. Here, there are movements of DNA from one chromosome to the other. This is actually the most common type of mutation seen in hematologic malignancies. A gain or loss of chromosome, that's also known as aneuploidy, and other types of mutations, which include gene deletions, point mutations, gene amplifications, and dysregulation of regulatory genes. Most leukemias need multiple mutations for the disease to be expressed. Some leukemias only need one or a single mutation for disease expression. For example, chronic myelogenous leukemia, CML, and acute promyelocytic leukemia. Both of these leukemias have one consistent translocation, which is enough for the development of the leukemia. We will talk about CML and APL in later chapters. Now let's talk about gain of function mutation versus loss of function mutation. Again, these are the types of mutations that you may find in leukocyte neoplasms, either gain of function mutation or loss of function mutation. So gain of function of a gene or loss of function of a gene, depending on the mutation. Gain of function mutations involve the activation of a proto-oncogene into an oncogene, resulting in increased signals for cell proliferation or inhibition of apoptosis, which is programmed cell death. Proto-oncogenes are present in normal cells, and they play a very important role in signaling pathways, cell proliferation, differentiation, and apoptosis. Proto-oncogenes are human genes that, when mutated, they have oncogenic potential. Some examples of proto-oncogenes are protein kinases, membrane signal transducers, nuclear transcription factors, and growth factors, also growth factor receptors. Gain of function mutations are considered dominant, and that is because because only one copy of the altered gene is needed for expression of the abnormal cellular function. So there are two general mechanisms of proto-oncogene activation. There's the qualitative mechanism and the quantitative mechanism. The qualitative mechanism involves structural changes in a proto-oncogene that result in the expression of abnormal fusion protein product. So basically, genes on different chromosomes are translocated, forming a new fusion gene that leads to the production of an abnormal protein product with abnormal activity. The quantitative mechanism, on the other hand, involves the dysregulation of a proto-oncogene that results in overexpression of its normal protein product. What this means is that genes on different chromosomes are translocated so that the normal gene is next to the proto-oncogene. This newly formed position on the same chromosome now leads to the overexpression of the gene with increased production of normal protein products. So again, in gain of function mutation, there are two common mechanisms, the qualitative mechanism and the quantitative mechanism. And again, here is a diagram that explains both mechanisms, the quantitative and the qualitative mechanism. Here in this slide are some examples of qualitative mechanisms in the activation of proto-oncogenes. Due to these translocations, there is production of abnormal fusion protein. So let's take a look at the first type of leukemia, CML, chronic myelogenous leukemia. The translocation that is involved here is the 922 translocation, which is designated as T922. So the two genes that are involved here are the ABL1 gene and the BCR gene. The ABL1 gene has tyrosine kinase activity, and the BCR gene on chromosome 22 is also involved here. This translocation results in a new fusion protein, which is called the BCR-ABL1 fusion gene, which is an abnormal tyrosine kinase that is constantly activated. It disrupts signaling pathways for cell proliferation, apoptosis, and DNA repair. In APL, the second leukemia that you see right here, the translocation that is involved in this leukemia is translocation 1517. And the genes that are involved are the PML gene on chromosome 15, which binds DNA. It's needed for cell proliferation, and the RA gene on chromosome 17, which is a transcription factor. The translocation leads to a new fusion protein, PML-RA gene, that blocks myeloid differentiation at the promyelocyte stage. The next leukemia is acute myeloblastic leukemia. So the translocation involved in this type of leukemia is translocation 821, and the genes involved is the RUNX1T1 or the ETO gene on chromosome 8, which is a transcription factor, and the RUNX1 AML gene on chromosome 21, which is also a transcription factor. After translocation, there is a formation of a new fusion gene, RUNX1T1, RUNX1, which is also known as the AML-ETO gene, which represses transcription of hematopoietic target genes of the RUNX1 in the core binding factor. This fusion now leads to a block in differentiation. We will definitely talk about all of these translocations when we start talking about acute leukemias and the ones that involve these translocations. And there are certain mechanisms that we will be talking about. The last one that is displayed here is the B lymphoblastic leukemia lymphoma, and the translocation involved is translocation 1221, and the genes involved is the ETV6, the TEL gene on chromosome 12, which is a transcription factor, and the RUNX1 or the AML1 on chromosome 21, which is another transcription factor. After translocation, the new fusion protein is the ETV6 RUNX1 fusion gene. And this fusion protein, this newly formed fusion protein, represses transcription of hematopoietic target genes of the RUNX1 in the core binding factor and leads to also a block in differentiation. In this table, we are showing you some examples of the quantitative mechanisms of proto-oncogene activation, which lead to an overexpression of the normal gene. For example, the first lymphoma that you see listed is Burkitt's lymphoma. The translocation 814 and the genes that are involved here are the MYC gene on chromosome 8, which is a transcription factor, and the IGH gene, immune globulin heavy chain gene, which is located on chromosome 14. This is a chromosome enhancer. This translocation leads to the overexpression of the MYC gene and dysregulation of cell proliferation. In follicular lymphoma, the translocation involved is 1418, and this involves the IGH, the immune globulin heavy chain gene, a heavy chain enhancer, and also the BCL2 gene on chromosome 18, which blocks apoptosis. The translocation leads to the overexpression of BCL2 and the inhibition of apoptosis. Lastly, in mantle cell lymphoma, the translocation is 1114, translocation 1114. The genes involved here are the CCND1 gene on chromosome 11, which regulates cell cycle, and the IgH, or the immune heavy chain gene, immune globulin heavy chain gene on chromosome 14. The translocation leads to the expression of the cyclin D1 and dysregulation of the cell cycle. Loss of function mutation, in order for loss of function mutation to occur, there needs to be an inactivation or loss of both alleles of the gene. And I'll talk about that in the next slide. Loss of function. So for this particular type of mutation that is seen in hematologic conditions, the mutation that is seen here is a loss of function mutation. Again, what it means is that there is inactivation or loss of something, some type of function. And it needs a mutation of both copies of the genes for the expression of the abnormal cellular function to occur. And some of the mechanisms involve inactivation of tumor suppressor genes, gene deletions, gene repressions through dysregulation of epigenetic mechanisms. Here are some therapies that are used in leukocyte neoplasms. We will definitely talk about these in more detail once we get to the chapters. So there are various therapies that are available for patients with leukocyte neoplasms. And the therapies can be divided into these categories. Chemotherapy, which is using agents. The only downside to this is that the chemotherapy agents affect both neoplastic and normal cells. And the effect is more profound on rapidly dividing cells, such as those of the mucosa of the gastrointestinal tract and also in the bone marrow. Some examples of chemotherapy agents include alkylating agents, plant alkaloids, anti-tumor antibiotics, anti-metabolites, glucocorticoids, and others. In the process of chemotherapy, there are 3 stages. Stage 1 is remission induction. Stage 2 is the intensification or consolidation therapy. Stage 3 is the continuation and maintenance therapy. Radiation therapy, okay? Radiation kills the cells by producing unstable ions that damage DNA and may cause instant or delayed death of the cell. The next type of therapy is supportive therapy. These are substances that are naturally produced that are now being synthesized and cloned in laboratories. For example, colony-stimulating factors are cytokines that normally act in the bone marrow to stimulate blood cell production. The same goes for erythropoietin. Erythropoietin, or EPO, is the main stimulatory cytokine responsible for red cell formation. Granulocyte colony-stimulating factor and granulocyte macrophage colony-stimulating factor have also been used to expand the number of mature neutrophils that are able to fight infection in the body. Again, these are agents that are known as supportive therapies. Targeted therapy is also something that is used in fighting leukocyte neoplasms. Because scientists and researchers have been able to identify specific genetic abnormalities that cause hematologic cancers, they are also able to identify targeted therapy that acts specifically on the tumor cells, leaving the rest of the cells untouched. Some examples of targeted therapy include imatinib, mesylate, or also known as Gleevec, which is used in CML. Imatinib is a tyrosine kinase inhibitor that inhibits kinase activity in all proteins that contain ABL, the ABL protein. Another example is all-trans-retinoic acid, also known as ATRA. This is another type of targeted therapy that is used in acute promyelocytic leukemias. Monoclonal antibodies such as rituximab target the CD20 antigen that is seen in non-Hodgkin's lymphoma cells. Also, hematopoietic stem cell transplants are obviously very difficult to perform. They are expensive and rigorous treatments, but they are alternative for patients with hematologic conditions. Some examples of those are some examples include allogeneic transplantations, which means from a donor genetically different from the patient, usually a sibling with matching HLA haplotypes. Also, there is autologous transplantations, and that's using the patient's own marrow or peripheral blood stem cells. In autologous transplantations, the cells are harvested from a patient after conditioning, and they are transplanted back to the patient. So, to conclude this short presentation, leukemias are going to be studied next. And so it's going to be very important for us to separate the leukemias. This table here specifies the two types of leukemias, acute and chronic. Acute leukemias are rapidly progressive and also fatal in weeks or months if left untreated. The white blood cell counts in acute leukemias could be increased, decreased, or normal. The symptoms typically include bleeding because of the low platelet count, fevers because of infection and because of the at times low neutrophil counts, and fatigue because of the low hemoglobin. Chronic leukemias are characterized by the development and accumulation of mature and maturing cells of a specific lineage. Chronic leukemias are less aggressive, and the survival rates are longer if left untreated. The white blood cell count in chronic leukemias is usually markedly increased, and the symptoms are variable. And thank you for listening to this lecture, and please move on to listening to the cytogenetics, the molecular diagnostic methods, and also the flow cytometric methods of white blood cell abnormalities.